Warm greetings to our evaluation lecturer, Miss Munira, my fellow friends. I am Narmata Valiappan. So today I would be presenting my experience at Veterinary Research Institute as an industrial training student. So without further ado, let's get right into it. So let's take a look at the objectives of this industrial training. The objectives would be to provide experience of the working environment to students in related fields with their specialization. And next, to train students to communicate professionally with employers and other staffs. Next, to enhance the ability to improve students' creativity skills and sharing ideas with each and other. Last, to allow students to get exposure to the latest systems. Let's take a look at history of the institute. So this VRI is formerly known as Federal Research Lab, which is established back in 1948. It was located temporarily at Hospital Bahagia, Tanjung Rambutan, Perak for veterinary research intentions by the courtesy of Director of Medical Services. In 2008, VRI was proposed by FAO, international bodies, to be recognized as a regional reference laboratory and for HPAI, highly pathogenic avian influenza A and Newcastle's disease. Now, Veterinary Research Institute was tasked to play a role in achieving the objectives of research to improve the productivity of the livestock sector through disease research and veterinary research development of vaccines and biological products based on the livestock. Research products produced such as diagnostic kits, vaccines, antigens or antibodies will be also direct impact on the industry, and it can be marketed locally and internationally through commercialization activities, which is transferring and selling out their products to other companies. So for the corporate, as for the vision, this is to provide reliable and efficient diagnostic services to the livestock industry for the prevention, control, and eradication of zoonotic, endemic, and epidemic diseases that impede animal health and production, public health, and the trade of the country. And for the mission, they are committed to support the growth of the animal industry by providing excellent service in diagnosis, control, and prevention of animal diseases through providing quality disease confirmation, advisory, and monitoring services performance by skilled individuals with high level of expertise using up-to-date facilities in the labs. So for about the department, I was placed under Parasitology and Hematology department. There are 15 sections and departments in VRI. So this section conducts diagnostic tests associated with parasites and blood parasites. Among the tests carried out in this section is Helminth's detection and numeration, detection of worms and blood-bound parasites in organs, and also blood protozoa in blood samples. So the types of samples that are handled would be blood, organs, skin, and feces. So for the types of tests that are done in our department would be first, McMaster, which is to determine the level of helminthiasis in animals by conducting the fecal egg count. This is done in feces sample. Next, formalin ether. This is to determine the types of parasite eggs present in an animal. Again, this is feces. Next, we have fecal culture. This is to obtain the type of level 3 larva for identification of worm present in an animal. Again, this is done in feces sample. Next, sedimentation to detect the presence of fluke eggs. Also, this is done in fecal samples on all the large ruminants like bovine. Next, one of the most important tests, which is thin blood smear, to detect the presence of blood parasites or protozoa in an animal. This is done on the blood sample of an animal. This is the daily tests that are done. This thin blood smear is the daily tests that are done in the lab. Next, we have Buffy code. This is to detect the presence of Trypanosoma species and Microfilaria species, which is a type of protozoa in an animal. Again, this is done in their animal's blood. Next, we have packed cell volume, which is PCV. This is to determine the percentage of red blood cell and white blood cell in an animal, which is done in a blood sample. Next, we have complete blood count, CBC, to measure the quantity of red blood cells and white blood cells in a sample, which is done in the blood sample. We have intestinal scraping to detect the presence of helminths and coccidia, which is done in intestine sample. And lastly, we have impression smear. This is to detect the blood protozoa in organ samples. These are done in organs such as lung, liver, spleen, and heart. So, as for the thin blood smear, the blood is taken from the sample with a capillary tube, a small capillary tube, and it is dropped on this slide, and the spreader is pushed forward. As you can see, the spreader is pushed forward along with the slide, and this will leave a nice thin blood smear. Next, as you can see, that's the smear that is formed on the slide. Next, we have Buffy coat. So same, we withdraw the blood with the capillary tube, and we would seal the end of the capillary tube with a sealant, and this sealant, sealed capillary tube, would be placed into a centrifuge machine for 5 minutes at 12,000 RPM. This will form a layer in between, which is called the Buffy coat. And further observations are done under the microscope. So if we scan the Buffy coat, we can see the blood parasites such as trypanosoma. And then this Buffy coat is observed by reading the level of it in a microhematocrit reader. So next we have McMaster. This is done by diluting the sample, the fecal sample of the animal, with mortile and pestle. The fecal feces is crushed, and it is sieved into, the filtrate is sieved into this counting chambers of McMaster slide. So once it's done inserting inside, you will see these examples of eggs under microscope, which is parasitic eggs. Next, we have simple flotation. The same filtrate is poured into a centrifuge tube, and a cow's lip is placed on top of it, the meniscus, for 10 minutes. After 10 minutes, the cow's lip is lifted onto a slide. It is lifted and examined under the microscope to see the presence of parasitic eggs. Now, lastly, we have impression smear. So this is like when a fresh cut is made on a portion of an organ with a pair of scissors. The tissue is then gently pressed onto a clean slide, and these slides are air dried and stained with Giemsa stain for microscopic observation. So the parasites would be stained. Let's take a look at the mini projects, interesting projects that I've done throughout this VRI journey. So first, we have this goat farm visit at Infoternak Sungai Siput, Perak. So in this mini project, we went to this goat farm, and that's the picture of it. There were like a lot of sets and species of goats, which they separated in different farms. So for the fecal samples, the feces are collected directly from the goat's rectum. A total of 27 fecal samples were collected fresh. A number of laboratory tests were carried out to identify the parasites and dominant type of worm in the samples, such as McMaster and fecal culture. So for the McMaster, all 27 goats were positive for strongyloides, and also 23 samples were positive for coccidiosis. And on the right, you can see the strongyle eggs that are found on the samples. We have strongyle on the left and fluke egg on the right. And for the fecal culture, after the observation, the majority of nematode in the fecal sample was Haemonchus contort, which is barber's pole worm. As you can see on the right, that's the image of Haemonchus contortus under the microscopic view that are found in the goat's fecal sample. So the next mini project would be at setting up a fly trap at Tambun Chicken Farm. So those are the pictures of setting up the fly trap. The second picture shows how you have to tie the knots on a pole on a steel rod under the ground. And as you can see on the right, that's after how I set up the complete net or the fly trap with a bottle on top. So the bottle needs to be empty with a hole for the flies to get inside and trapped completely. And in the middle, you can see how the flies are trapped in after 2 hours. These are the number of flies that are trapped in. So the aim of this project is to identify the majority species and sex of the flies at the chicken farm. Since it was a harvest season, there were no much chickens. So this led to a lesser number of flies in the whole poultry. However, we managed to trap about 240 flies in 2 hours. Those flies were stored overnight in the freezer to ease the counting process and further observations. The flies were transferred into petri dish to observe under the microscope and count them. And the pictures showed how the flies were counted under the stereo microscope. They were also observed under a stereo microscope to identify the species and sex. By observing the mouth and the distance between their eyes, it was concluded that all the flies trapped are house flies, which is Musca domestica, and all the flies are female flies. So the comments and suggestions that personally I would like to explain in this throughout my industrial training is that I feel like there was a minimal supervision for laboratory skills and activities that are done in the lab or at the field work. I feel like the supervisor should observe my laboratory skill and communicating skills and also supervise how I'm doing in these project works and field works before considering for grading the marks. Next, the students might need to learn more than in this department for 20 weeks. So 20 weeks is such a long time, and VRI should have a rotating system for students since there's like 15 sections and 5 units in the whole building. Lastly, bad facilities and uncomfortable workplace. So I feel like they should give more importance to students at a workplace with better facilities, because it's really hard to work under a very hot and uncomfortable situation wearing the lab coat and mask, especially the air conditioner in lab, because there was no good air conditioner in the lab, so it was really hard for ventilation. So I feel like they should focus more on how the students are doing in the lab. As for the conclusions, I've learned a lot throughout my internship journey. Not only the knowledge of technical skills and technology used in the science field, but I also learned how to communicate among the colleagues and how to manage the tasks and assignments given to be completed on time. I've also improved my communication skill, teamwork spirit, as well as learning about the work culture after several lessons. I've also learned how to work under pressure and respect the staffs and head of departments. Lastly, I'm really grateful for the new experience and opportunities to train myself for betterment. That's the end of my presentation. Thank you so much for lending your ears.